CRISPR-Cas system in bacteria provides adaptive immunity against invading DNA. Phage, plasmid. RT-PCR detects RNA by converting it to complementary DNA using enzyme, reverse transcriptase. GenExpert MTB-RIF Ultra increases sensitivity by amplifying multi-copy IS6110 and target IS1081. Loop-mediated isothermal amplification LAMP operates at a constant temperature of 60 degrees Celsius, 60 to 65. Next-generation sequencing platforms predominantly use sequencing by X chemistry. Synthesis. Recombinant subunit vaccines require adjuvant to enhance immunogenicity. AS04 is a LUM plus agonist. TLR4, MPL, monophospholipid A. mRNA COVID-19 vaccines deliver nucleoside-modified mRNA in lipid particles, nanoparticle LNP. ChAd-OX1 nCoV-19 vector is replication-deficient chimpanzee virus, adenovirus. Multi-epitope conjugate vaccine type BAR TCV uses Vi polysaccharide bound to protein carrier, tetanus toxoid. ECG revaccination is not recommended because it interferes with interpretation of a test. Tuberculin skin. Reverse vaccinology identifies vaccine targets using in silico analysis of gen genome, pathogen. Quadrivalent HPV vaccine targets type 6, 11, 16, 18. OVX836 is a nucleoprotein-based universal vaccine candidate against one virus, influenza A/B. Spatial transcriptomics allows mapping of gene expression in tissue sections using barcodes, spatially indexed positional. Metagenomic NGS in clinical microbiology primarily sequences conserved -RNA gene for bacteria, 16S rRNA. CAR T-cell therapy uses lentiviral vector derived from blank virus, human immunodeficiency virus, HIV-1. First WHO pre-qualified malaria vaccine, RTS-AS01, targets circumsporozoite protein of Falciparum, Plasmodium. Immunobridging non-inferiority design compares neutralizing antibody geometric mean titer, GMT ratio, lower bound, greater than 0.67. Nanopore sequencing identifies nucleotide changes by measuring current alterations. Ionic, electrical. Dry swab direct RT-PCR bypasses step, reducome turnaround time for SARS-CoV-2, RNA extraction. WHO defined community SARS-CoV-2 CT as high viral load for infection control decisions. 30. DNA vaccines such as ZyCoV-D use intradermal needle-free jet injector, spring-towered pharmajet. Antisense morpholino oligomers targeting Ebola, VP24, VP35, were branded as BRAIN, AVI 7537 PMO. Digital PCR quantifies absolute copy number without relying on curves. Standard/calibration. Serological surrogate of protection for hepatitis B is anti-HBs titer greater than MIU per milliliter. Viral neutralization by pseudovirus assays replaces pathogenic virus with reporter system. Luciferase, GFP. Droplet digital PCR partitions sample into only 20,000 droplets. Endpoint fluorescence allows ratio, positive-negative, Poisson. Genome editing therapies use high-fidelity CRISPR-Cas9 variants to minimize cuts. Off-target. Speak Illispot. Measures cytokine spots released by lymphocytes after antigen stimulation. Break time 400 milliseconds. If N-gamma interferon gamma, prosody. mRNA vaccines incorporate N1-methyl pseudouridine to reduce innate immune activation via sensors, TLR7, 8. Bivalent Omicron booster uses ancestral Wuhan strain plus spike from BA4/sublineage, BA5. Gavi COVAX facility accelerates equitable access to vaccines worldwide. COVID-19. E-specific antibody emicizumab for hemophilia A mimics factor VIII by bridging FIXA and X, FX, factor X. In spray intranasal COVID vaccine, Incovac uses non-replicating vector, adenovirus type 5. Sanger sequencing chain terminators are dideoxytriphosphates, nucleotide DDNTPs. qPCR efficiency ideal value is 90% corresponding to slope minus 3.32, 100. MALDI-TOF MS uses matrix alpha-cyano-4-hydroxycinnamic acid and measures charge ratio, mass. Vaccine adjuvant CpG 1018 is synthetic dinucleotide that activates TLR9, unmethylated. BioFire film array multiplex PCR panel delivers syndromic pathogen identification in minutes, 60, about 1 hour. Lyophilized vaccine vials must avoid repeated cycles to maintain potency. Freeze, thaw. WHO pre-qualified oral cholera vaccine Shanchol contains killed Vibrio 01 and serogroups, 0139. Needle-free jet injectors reduce Sharp's injury but risk cross-contamination if not disinfected. Nozzle/jet tip. VTT conjugate typhoid vaccine provides protection for at least 5 years per trial data. mRNA 1273, Moderna dose is 100 micrograms mRNA in 2 ml volume IM. 0.5 ml. Helicopter sequencing Hi-Fi CCS by PacBio generates long reads with accuracy greater than 80%, 99.9. SNP genotyping of MDR-TB cluster isolates by Miro VNTR uses loci for standard comparison. 24. eTest is an Auger gradient diffusion method providing direct value, MIC, minimum inhibitory concentration. Recombinant zoster vaccine uses VZV glycoprotein E combined with AS01B. Schedule 2 doses, 0 and 6 months, 2 to 6 months. BCG Danish and Tokyo substrains differ in RD1 deletion. Vaccine efficacy variable due to blank genetics. Host, HLA, IFN gamma. SARS-CoV-2 lineage nomenclature Pango uses a letter followed by numbers. A, B, then sublineage. Example, B11. Oxford nanopore minion flow cell contains 400 nanopores, 512 approx. 2048 in R10.4. DeepMind's AlphaFold predicts protein 3D structure from WINI sequence, amino acid. Spatial clustering of measles cases is monitored using genomic typing N-gene, 8-segment sequence genotyping. WHO recommends fractional IPV, 0.1 milliliter intradermal, to stretch supply. Vaccine. Monoclonal cocktail Evusheld combines tixagevimab with, for COVID prophylaxis, silgavimab. Pentavalent vaccine combines DPT, Hep B, and conjugate. Haemophilus influenzae type B, HIB. SNO RNA-guided base editing is an emerging CRISPR approach using deaminase converting AT2 base pair, GC, adenine base editor. False negative rapid antigen tests occur at CT greater than H on RT-PCR for SARS-CoV-2, 30 to 33. Praziquantel mechanism discovered using cryo-EN shows binding to TRPM channel. Transient receptor potential melastatin, PZQ channel. mRNA vaccine stored at -20 degrees Celsius remains stable for 2 months, 6 months. Rapid Mini Sequencer uses isothermal RPA plus Oxford Nanopore for same-day Inquester ID, pathogen AMR. Serology cutoff index COI in chemiluminescent assays is ratio sample RLUs to RLUs. Calibrator control. Antigen processing in mRNA vaccines occurs via MHC class pathway to activate CD8 T cells. Class 1. Ebola ring vaccination strategy uses recombinant VSV vector expressing Ebola glycoprotein, Zaire GP. RT lamp visual readout often uses color change of pH indicator, phenol red. Protein-based nanoparticle vaccine, NVX-CoV2373 uses saponin-based matrix adjuvant, matrix M. One Health approach integrates human, animal, and health surveillance, environmental. RAT for Streptococcus pyogenes targets group A carbohydrate antigen detected by immunoassay, lateral flow. Sanger sequencing, read length, not BP contrasts with Illumina 200 BP and nanopore greater than 10 kb, 800 to 900. Hib conjugate uses polyribosol ribitol phosphate linked to protein, tetanus toxoid, CRM197. mRNA vaccine cold chain breach is indicated by color change of vial monitors, time temperature indicator, TTI. Expert XDR cartridge detects resistance to INH, fluoroquinolones, and ethionamide. Second-line injectables, amikacin, kanamycin. WHO pre-qualification requires vaccine lot-to-lot consistency within N times standard deviation, plus minus 1.5. Metagenomic pathogen detection analytic parameter LOD defined as 100 copies per mil. Limit of detection, variable 10 to 100. E. coli water test detects E. coli by fluorescence of enzyme, beta-glucuronidase, MUG. OPV, 0 dose at birth primes gut immunity. Subsequent switch to BOPV removed X serotype. Type 2. In CAR T therapy, cytokine release syndrome graded equal to 3 is treated with IL-6 blocker, tocilizumab. Digital ELISA Simoa achieves femtomolar sensitivity by isolating single cells in microwells. Enzyme-labeled immunocomplex. qPCR absolute quantification utilizes standard curve. Relative quantification uses delta-delta absolute method, CT. Gene-drive CRISPR mosquitoes aim to suppress aegypti population controlling dengue. 80s. Nanobodies are single-domain IgG derived from antibodies. Camelid, Llama. Phase 4 post-marketing vaccine surveillance is also called monitoring. Pharmacovigilance, safety. Fluorescence in situ hybridization (PNA) fish detects Candida albicans directly in positive culture, blood. SARS-CoV-2 subgenomic RNA detection indicates active replication because sgRNA has leader sequence, 5-leader TRS. Holio environmental surveillance tests sewage using virus concentration by two-phase separation, PEG, dextran. Epivac corona peptide vaccine relies on synthetic SARS-CoV-2 spike epitopes conjugated to carrier protein, virus-like particle, chimeric protein. Pan-fungal PCR targets conserved rDNA ITS2 region, internal transcribed spacer. Single-cell RNA-seq libraries prepared using 10x genomics partition cells in single-cell RNA-seq droplets, gel bead in emulsion, GEN. Dedicated mRNA cell-free vaccine platforms enable rapid candidate production in under days, 30 days. RT-RPA, recombinase polymerase amplification, operates at 2 degrees Celsius, enabling field diagnostics, 37 to 42. Speak, prosody rate 120%. Global polio eradication endgame strategy includes introducing IPV to mitigate risk of virus. Break time 400 milliseconds, CVDPV. Vitas Brahms PCT measures procalcitonin, cutoff equals nagrams per milliliter, suggests severe bacterial sepsis, 2 nagrams per milliliter. N-chip microfluidic card performs HIV syphilis duplex ELISA using question change readout, colorimetric. Cholera rapid dipstick crystal VC detects O1 and O139 LPS. False positives avoided by testing DASH colonies. Enriched broth 6H. Fluorescent in situ sequencing FISSEQ can map viral integration sites in Gemin's genome. Host, human. Zika congenital syndrome risk highest if maternal infection in the trimester. First. Enterovirus D68 outbreaks monitored by real-time RT-PCR targeting prime range region, UTR. Adjuvants inducing Th1 bias include synthetic double-stranded RNA, agonist, poly-IC, TLR3. ACIP now recommends PCV15, followed by PPSV23, or single PCV20 for adults equals 65. Prosody rate 20%. Lyophilization curve endpoint TG represents glass transition temperature critical for stability. Vaccine or biologic.